talking about our study on kelp forests and their community structure. We conducted our study on the west end of the San Nicolas Island in California. We decided to conduct our study here because we noticed many barren areas where kelp forests were not growing. In order to properly analyze and understand our study, I will introduce some basic concepts on kelp forest community structure. Kelp forest communities have plant and herbivore interactions. This is a very important element in determining why an area has gone barren and why some areas continue to have a thriving kelp community. Regardless of location, all kelp forests are susceptible to the following: sea urchin grazing and low recruitment in substratum. What are barren grounds? They frequently occur adjacent to ungrazed areas. How do they occur? The transition from a kelp forest to barren ground typically involves the changes in feeding behavior of sea urchins. This feeding mechanism change is independent of sea urchin density. What is the purpose of our study? To investigate the factors that affect the feeding behavior change of sea urchins and analyze each position in regulating kelp forest community health. How did we conduct our study? First, we determined the density of algae and urchins in barren and kelp forest communities and continuously monitored them over the time of this study. Second, to check the feeding behavior of urchins, we had multiple ways of assessing them. We assessed drift algae abundance, daily movement of the urchins, the diets of the urchins, reproductive health, and their microhabitat distribution from the barren and kelp areas. The study site was done by scuba diving in shallow waters. The water was around 11 meters in depth in the subtidal zone off the southwest end of San Nicolas Island, Ventura County, California. Some descriptions about the study site. The bottom of the subtidal zone consists of sandstone and shale pavement cut by numerous crevices and ledges and large boulders. We set up 2 sampling stations, one in a barren area and one in a kelp forest adjacent to the barren area. Each area was established with 50-meter parameters. Our first observation of each area are as follows. The barren area consisted of many encrusting coralline algae. Brown algae was non-existent. Sea urchins were abundant and grazing the substratum. The kelp areas had a surface canopy with lots of brown algae. Encrusting coralline algae was also found but not in as large of numbers as the barren area. Sea urchins were abundant but preferred to live in cracks and pockets, which proposed us to think that the sea urchins were feeding on drift algae. To determine abundance of drift algae, we took samples from 10 randomly placed 10 square meter plots inside of our barren and kelp study areas. Drift was placed in bags and taken to the laboratory. We established 2 more sites to measure the grazing patterns of the sea urchins. We also measured daily movements in this area, and we tagged 25 urchins by slipping short straws over their spines.
Reproductive health, we measured gonad indices of 10 randomly collected sea urchins from each site. After we brought them to the surface, we injected formaldehyde into animals through their peristomal membrane. We waited 24 hours and dissected them after, removed the alimentary tracts and placed them in jars for further analysis. Gonads were removed and dried on paper towels and weighed. Since gonads suffered fluid loss, we estimated the whole animal weight mass instead. For diet, we collected 19 urchins from both habitats and took them to the lab. Urchins were dissected and stomach contents were washed out and separated by allergy type. The algae in the stomach were washed with fresh water and placed into a beaker. To measure microhabitat distribution, We tallied urchins live in cryptic or urchins live in open habitats. The formation of the berry site changed substantially during our 2.5-year study. The kelp site, on the other hand, remained the same throughout the study. For the berry site, densities of brown algae were lower at the start of the study. Over time, the density of brown algae increased, and near the end of our study, the density of each site was similar. As for the urchin density, the berry site showed fluctuating population of urchins, whereas the kelp site showed a slight increase. As we have mentioned before, urchin density played no impact on the urchin grazing activity. Next, we will talk about the recruitment of young algae in each site. Berry site showed increase in density of small brown algae, but then decreased. Only the large plants that survived increased in density. This boundary is formed from high mortality of young algae and plants that are able to survive urchin grazing. Kelp site showed less increase in density of brown algae. This less increase suggests that young recruits were able to survive and grow to adults. The abundance was influenced by both surrounding flora and the time of the year. In the kelp site, algal biomass was high, which meant drift biomass was high as well. The drift consisted of around 87% to 98% brown algae. Coralline algae was not as present. Drift biomass was high during the summer when the water motion was long. Drift biomass was long during the winter and early spring when water motion tends to be higher, and drift was also being moved due to the stronger water movement. In the berry site, algal biomass was long, which corresponded to drift abundance being low. The berry site also showed much more coralline algae. For urchin movement, we can see many clear differences between the two sites. In the berry site, urchins showed more movement than the kelp site because of lack of the drift algae. As drift abundance increased throughout the study, movement rates decreased in the berry site. From our observation, we can conclude an inverse relationship between urchin movement rate and drift abundance. The diets of urchin from both sites composed of mostly brown algae. The berry site, however, showed signs of red algae. By the end of the study, both of the diets of urchins from each site were indistinguishable. We can make some assumptions from our observations. The berry site urchin's diet probably consisted of young brown algae recruits that drift. Gut contents of other urchins without young algae recruits showed coralline algae consumption. Kelp site urchin diets more than likely consisted of drift since the abundance of algal drift was high. Grazing activity was also long, which plays another factor in determining diets of the urchins.
In terms of the gonad indices, the berry site urchin gonad indices were at the start lower than the kelp site. By the end of the study, the berry site urchin gonads had increased to a similar value as the kelp site urchin gonads. However, both origins from each site decreased during spanning the spring. Gonad size also a good indication of the origin's well-being. Shows how much food is available. A difference in food availability can result in different sizes of gonad indices. This means that kelp site origins gonads were larger because of the food available to them, whereas the Balen site had smaller gonads. Microhabitat distribution was as simple as black and white. Origins either occupied open habitats or cryptic habitats like cracks and pockets. Different feeding behaviors are directly correlated with microhabitat distribution. Seeing a shift of origins from a barren area to cryptic habitat resulted from more drift algae being present. The reverse is true as well. If origins are seen to be shifting to grazing, this means that there is less algal drift present. We tested this idea in the field as well. We set up a 40 meter by 40 meter plot near our site, cleared brown algae and monitored origins before and after removal of algae. At the time of removal, 89% of origins occupied cryptic habitats. After coming back after 6 months, we can see that less than 50% of origins remained in cryptic habitats. To conclude our study on the kelp forest community on the west end of San Nicolas Island, we would like to discuss our study as a whole. First, our system is simple. Look at the barren area and kelp area and see how that changes over 2.5 years. Second, we analyzed origins and their behavior. Origin densities in the two sites were similar and remained the same throughout our study. For the kelp-dominated area, we saw that sea origins took no part in grazing due to large abundance of algal drift. For the Balen site, there were many issues that we observed. Drift algal was no insufficient supply to feed the origins. This resulted in origins activity grazing the substratum. This prevented establishment of recruit. However, some plants were able to survive, and once they started producing drift algae, origins began behaving like the kelp site origins. This transition of origin behavior allowed the kelp forest in the Balen area to establish itself. Our study shows that origins maintain Balen areas by eating all the young algae and not allowing them to grow into adults. Our study also showed that drift algae is important in preventing grazing of the sea origins. To summarize the kelp forest communities, we propose a qualitative model to show the relationships in the community. The relationships are as follows. Algae density in Balen area was low, which means low drift abundance. Low drift abundance means low nutrition of origins. Poor nutrition means that origins start grazing in order to feed themselves. Grazing led to barren areas. In kelp-site, drift was abundant, which means origins were well nourished and no grazing occurred. The big question is, how do these habitats become barren? Our study does not address this directly, but we can suggest a theory as to how it may occur based on our study. Kelp forest would thrive if there were no outside forces to agitate them. How they become disturbed may be due to plants being removed from storms and strong currents which may latch onto other plants and drag them away along with the drift. The opposite effect may take place as well, where a barren area is supplied with many nutrients and goes undisturbed, allowing a kelp forest to thrive. So through our experiment, we have identified important aspects of the kelp forest community structure and the relationships that exist within the community.